Soon you will start the process of working with bacteria. In some ways, it is like planting a garden, as you will place your microbe into a source of nutrients that we call a growth medium. These nutrients will provide the materials needed to support growth of the microbe so that it can be cultured and studied. Bacterial growth media can be liquid or semi-solid, nutritionally rich or poor, modified by the addition or removal of key nutrients, all in an attempt to give us the tools needed to find out more about our unknown microbe. Here are some terms and definitions and facts about media. Liquid media are called broths. If you dissolve nutrients into water, you have created a broth. They are mostly water, and many broths are over 99% water. Broths normally are dispensed into culture tubes and then autoclaved to sterilize the contents prior to use. Liquid media can be converted to semi-solid media by the addition of 1.5% agar. Agar is a seaweed extract that dissolves when boiled and solidifies again when its temperature drops below about 45 degrees C. It has the consistency of gelatin, but media containing agar are still often up to 97.7% water. Agar provides no nutritional content and is not digested by normal bacteria, making it a great solidifying agent that is not destroyed by bacterial growth. When agars are used to make semi-solid media in Petri plates, they are autoclaved in flasks and then dispensed into sterile plates and allowed to solidify. Agar plates are useful for separating and purifying bacteria by means of streak plating, which you will study soon. When you want an agar medium in a tube, the medium is boiled in a flask to dissolve the agar, and then it is dispensed into tubes before being sterilized with the autoclave. After autoclaving, the tubes are allowed to solidify prior to use. If the tube is allowed to solidify normally, the tube is called an agar deep. If the tube is allowed to solidify in a slanted position, it is called an agar slant. Slants and deeps are useful for some biochemical tests. Media can be modified to limit which bacteria can grow on them by removing key ingredients or adding chemicals called inhibitors that block the growth of unwanted bacteria. These media are called selective media. Media can also be modified so that it is easy to tell whether the metabolism of a microbe has produced a specific product by adding chemicals called indicators. Two bacteria with different abilities to perform the reaction would produce mature cultures of different colors. These media are called differential media. Sometimes enrichments are added, like blood or other additives with exotic nutrients not found in normal media. Such media are called enriched media. Some bacteria are very picky eaters. We call them fastidious organisms. And without enrichments, we would not be able to grow them on plates in the lab. So it is safe to say that a fundamental part of our study of bacteria in this course will center around the variety of growth media used for their growth and study. We will look at the types of media mentioned here in greater detail as we move through the semester.